Good afternoon, everyone. This is Dr. Rasmi. Today we will discuss about to the topic. Already last class to complete the male gametophyte development. What is the male gametophyte? These are things are completed. Now we will start to the female gametophyte. Female gametophyte. What is the female gametophyte? How to female gametophyte is used for the things are these are things are yesterday already explanation is completed. Now we are going to explain today megasporangium already completed. Types of embryos we will discuss about to the class. See here, types of embryo sac. The three types of embryo sac are represented by, that is the diploid, the polygonium type of three type of embryo sac. One is the monosporic, second one is the bisporic, third one is called the tetrasporic embryo sac. Here, the polygonum type. First one is the monosporic. The polygonum type of embryo sac is described above. The above is formed from one of the four haploid megaspore nuclei. One of the four megaspore nuclei, which in turn formed formed from diploid, which is turned from formed from diploid condition megaspore mother cell as a result of meiosis, although it is the most common type of mode of embryo sac development in all angiospermic plant. Even there is a substitutional number of plants where more than one megaspore nuclei take part in the take part in the process. Therefore, depending on the basis of involvement of number of megaspore nuclei, in its formation different type. One is the monosporic, second one is the bisporic, third one is the tetrasporic. First one to explain about to the monosporic embryo sac. Monosporic embryo sac is the one where only one of the four megaspores take part in its formation, as in the polygonum type. This is called the polygonum type. Here, monosporic again, the depending upon the number of nuclei, this is can be divided into two types. One is the polygonum type. Second one is the enothera type. Polygonum type, 8 nucleate structure is formed, and four nucleated structure is called the enothera type. Therefore, there are three of the megaspores, usually those which are at micropylon, degenerate, leaving only one functional megaspore. In this type of embryo sac, all the nuclei are genetically identical because they are formed through mitosis of the single nucellus. Monosporic embryo sac further divided into polygonum type, enothera type. Polygonum type is called the 8 nuclei structure. Enothera type is the 4 nuclei structure. Polygonum type 8 nucleate, as described earlier, it is formed by the chalagal megaspore of the tetroid, and is the 8 nucleate. Tetroid is the 4 double, that is the total is the 8 nuclei structure is present. The mature polygonum type of embryo sac compresses three egg cells, three antipodal cells, two binucleate central cells. 3 plus 3 plus 2, 3 plus 2 plus 3. 3 is the egg apparatus, 2 is the binucleate, 3 is the antipodal cells. This type of embryo sac is the most common and is therefore commonly digestinated as the normal type. However, it is also designated as the polygonum type because it was reported the First reported the plant name is called the Polygonum divaricatum. See, after mitotic division, here see four cell structure to form. After division, eight cells. In the eight cell, the final, this is a polygonum type of embryo, sir. The upper part, these three cells name is called the, three is the antipodal. This one is the, three cell is the egg apparatus, and this is the antipodal cells, egg apparatus, antipodal cells, binucleated type. The second one is the Enothera type or four nucleate type. Enothera type of embryo sac is derived from the micropyle megaspore. Micropyle megaspore of the tetrad and is four nucleate. The mature embryo sac consists of an egg apparatus and a uninucleated central cells are present. This type of Enothera type of embryo sac is found in the Oraganaceae family. Oraganaceae, where you can observe Oraganaceae family. In Enothera lamarckiana, the embryo sac is usually formed by the micropyle megaspore. This type of embryo sac usually, what type of Enothera lamarckiana type of embryo sac? This type of embryo sac usually formed by the micropyle megaspore of tetrad. These which undergoes only two nuclear, in this part only divided into two nuclear division instead of the usual three occurring in the polygonum type of embryo sac. In this way, four nuclei are produced which organize into the two synergid cells, the egg and a single polar nucellus. Since the third division is omitted and all the nuclei are situated in the micropyle part of the developing of the embryo sac. There is neither a lower polar nuclei nor antipodal nuclei. See, this is the Enothera type. The divisions is the this one. Here, three polar nuclei are there. Lower polar nucleus or antipodal, any antipodal cells are there. Total four nucleated cells are present. Next, the second one is the bisporic bisporic embryo sac. Bisporic embryo sac development. No tetroid in megaspore development. Here, the second one is the bisporic. Monosporic is completed. Monosporic, two types of monosporic. One is the polygonum type, 8 nucleated structure, 3 plus 2 plus 3. The second one is the monosporic and polygonum. Second one is the Enothera lamarckiana polygonum example. The second embryo sac is called the second one is the embryo sac Enothera type. The coming to the second one, bisporic embryo sac. Bisporic embryo sac development not treated in megaspore development. Here, megaspore development is not treated in the no tetroid in megaspore development. In the bisporic embryo sac, tetroid megaspore is not development. From the dyad degenerates and the other develop into embryo sac. Here also two type. One is the allium type. The second one is the endamine type. Two types are there. Allium type, endamine type. Here no tetroid in megaspore. In the megaspore, tetroid structure is absent. Development of the megaspore. From dyad only degenerate. Dyad structure only degenerate and other development into the embryo sac. Again, this one bisporic embryo sac also two types. One is the allium type. The second one is hello, hello, hello. చెప్పండి. నేను క్లాస్లో ఉన్నా, మళ్ళీ చేస్తా. The second one is the endamine type. Allium type reported in Allium fistulosum by Strasburger. Allium type reported by Allium fistulosum. Allium fistulosum by Strasburger scientist in 1879. This type of embryo sac in the bisporic embryo sac again to divided into two types. One is the embryo sac. One is called the allium type. Another one allium type is reported. The plant name is called allium fistula. Fistulosum. Allium fistulosum. By this one is this one is the example for this one. Explanation. The scientist name is called Strasburger in 1979. The Strasburger scientist to explain this one in 1979. It from it one from it is called it by three mitotic division. You can observe here three mitotic divisions are present. This is the 8 nucleate embryo sac is formed and later organized. Hmm, Organized to cells structure. To organized to seven cell structure. See, this is the divisions. Here you can observe after divisions the cells 4, 8, 3 plus 2 plus 3. Total 3 plus 3, 6, 7, total 8 cell structure. And coming the second one is endaminotype. Endaminotype, this is the reported hispanicus. The plant name Endamino hispanicus. Endamino ipscascum from by three mitotic by from three mitotic from it, three mitotic divisions are there. In these divisions, eight nucleate embryo sac is formed and later organized seven-celled structure. Here you can observe to the in this part only seven-cell structure is present. Endamine type. See types of embryo sac development: monosporic and bisporic method. See, this is the female gametophyte embryo sac development. Megasporogenesis and megagametogenesis. Both are there. Both are present. Megasporogenesis and megagametogenesis. Megasporogenesis, monosporic embryo sac. Monosporic embryo sac, polygonum type, and megasporomiasis 1, 2. This is the total mega monosporic embryo sac, polygonum type. To final 3 plus 2 plus 3 is there. First megaspore to divide meiosis 1. First meiosis 1 to division 2 nucleus to form. Second meiosis 4 nucleus. This is the sporogenous megasporogenesis. When the megagametogenesis, the divisions is the total meiotic 1 to form the 4, 1, 3 plus 2. Meiotic 2 is the 4. Meiotic 3 is the 3, 4, 8 new cells. This is the mature. This is the monosporic 4 nucleate Enothera type. And this one is the bisporic 8 nucleate allium type. 3 plus 2. Just like the this bisporic and polygonum type, both are same, similar. 8, 3 plus 2 plus 3. Bisporic 8 nucleate endamino type, this one. The amino type. Coming to the last one is the tetrasporic embryo sac development. Tetrasporic embryo sac development, the meiotic division. The meiotic division of the MMC. MMC meaning megaspore mother cell. Megaspore mother cell is not followed by cytokinesis. The megaspores mother cells is, this is the not connected, cannot followed by the cytokinesis. All the four nuclei take part in embryo sac formation. The development of embryo sac from the four nucleated type, that is the cenomegaspore, is complicated, and the further divisions vary in different species based on the arrangement of nuclei post mitotic division in the cenomegaspore and the presence or absence of the nuclear fusion. These are the various type of recognized in this type. The first one, two major types, which are subdivided again into seven types. First two major type, first one is those without nuclear fusion. Second one, those with the nuclear fusion. See, this heteroscopic embryo sac development without nuclear fusion, some two are there. One is the without nuclear fusion. Second one is the with nuclear fusion. Without nuclear fusion again can be divided into five types. Two 8 nucleate and three 16 nucleated. See, in this one again this without nuclear fission can be divided into five types. In the five, stages is the 8 nucleated stage. Remaining 3 is the 16 nucleated. Adaxo type 8 nucleate. Plumbago type 8 nucleated. Pina type 8 nucleate. Drusa type 16. Pina type, sorry, 16 nucleate. Drusa type 16 nucleate. Peperomia type 16 nucleated. This is the without nuclear fusions. Next one, 8 nucleated without nuclear fission. The first one, Adaxo type, 8 nucleated structure, type of embryo sac, reported in Adaxo mosaiellina by Johnson scientist, Johnson scientist in 1880. The four nuclei after one meiotic division organized into three-celled egg apparatus, two nucleate central cells, three antipodal cells. See, this is the same like that with polygonum type 3 plus 2 plus 3. Next one, the second adaxon. Next one, plumbago. 8 nucleate. This is the tetrasporic embryo sac development. 8 nucleated without nuclear fission. That without nuclear fission, that one second one is called the plumbago type. Here, observed in the plumbago comparisons, companisons, plumbago companisons. The plant name is called Plumbago campanices. Out of the four nuclei, after one mitotic division, one uninucleate egg cells. Here, four nuclei structure after one meiotic division, one nucleate egg cells at four nucleate central cell and three nucleated peripheral cells are formed. And the next one is the without nuclear, with nuclear fusion is called. Next one, third one, tetrasporic embryo sac development, the 16 nucleated, 16 without nuclear fusion. First one is the Pina type, second one is the Plavago type. The first one is the phenotype. Phenotype reported in the Brachyspona and Sarcocola. These two plants you can observe to the Pina type of embryo sac. The four nuclei by the towards four nuclei by the towards four nuclei by the towards divisions 16 nuclei are formed. They are arranged in 3 plus 3 plus 3 plus 3 plus 4. The cells arranged into total 16. Uh, 3 plus 3 plus 3, 4. Here conditions as three-celled micropylar egg operators. See, this is the first one. Second, here total 16 cells. Three egg operators. 1, 2, 3. Three-celled chelazal antipodal. Two lateral cells. Lateral group of cells. Four functional polar nuclei. 3 plus 3 plus 3 plus 3 plus 4. 3, 3, 3, 3 plus 4. Total 16 nucleate structure, that is the phenotype. Next one, the second thing, tetrasporic embryo sac development into development 16 nucleate without nuclear 16 nucleate without nuclear form. This is the 16 nucleate without nuclear fusion. First one is the Drusa type. Observed in Drusa appositifolia. Drusa appositifolia. 16 nuclei are here also to 16 nuclei are present. This is 16 nuclei formed arranged as a three-celled egg operators. See, this is the three egg operators. These two polar nuclei, these all are 11 are called antipodal cells. 3 plus 2 plus 5 plus 11. Total 16 cell structure. 16 nuclei are formed in the Drusa apposifolia. Coming to the next one, Phenamia type. Reported Phenamia the without with nuclear fusion. Without nuclear fusion. The next one is the second one is the with nuclear fusion. Tetrasporic embryos ఇది చెప్పలే కదా without peperomia drusa, and fifth one is the peperomia. Reported in peperomia pellucida. Here 16 nucleated are formed arranged in the two-celled egg operators. See this one. These also after division to form to the 16 nucleated peripheral cells. You can observe to the 16th one is 6 nucleated peripheral. One is a formed two-celled egg 16 nucleated as the two-celled egg operators. These two egg operators, a central 8 polar cells, 8 plus 2, 10, and 6 nucleated peripheral cells, 6. 10 plus 3, 6 is the, these 6 is the nucleate peripheral cells. This is called the 3 plus 2 plus 8 plus 6. This one is the peperomia without nuclear fusion. Coming to the next one, tetrasporic embryo sac. Development 8 nucleate with 8 nucleate with nuclear fission. This type of embryo sac development 8 nucleated type with nucleus with nucleus with nucleus with nuclear fission. Out of the 4 nuclei, 3 fused to form a triploid and chelazal end. One remain as the haploid at the micropylon. 2 variations are reported. Here with the nuclear fission, total 8 nucleated structure with the nuclear fission. Out of the 4 nuclei, 3 fused to form a from a triploid nucleus at chelazal end. One remains as haploid at the micropylon. 2 variations are reported. For this one is the 2 types is there. One is the Fertilaria type. Second one is the Plumbago gella type. The first one is the Tetrasporic embryo sac development, 8 nucleate with nuclear fusion. Fertilaria type, the first one, present in the various species. You can observe to the Lilium species. In this type of tetrasporic development, where the Lilium species are there, in that Lilium species, the fertilaria type of embryo sac can observe. Here, both haploid and diploid nuclei, both nuclei, haploid, 2, triploid, 3 nuclei divide twice mitotically. Both nuclei divisions are there twice, 2 times and mitotically to form the 8 nucleated type of embryo sac. In the 8, 3-celled haploid egg operator, 3-celled triploid antipodal cells, and 2 polar nuclei organized. One polar nuclei is the haploid, other one is the triploid. See, 3-celled egg operators, 3-celled triploid antipodal, 2 polar nuclei are organized. One polar nucleus is haploid and other than is called the triploid position. The second one, tetrasporic embryo sac development, 8 nucleate with nuclear fusion. The second is the Plumbagella type. This Plumbagella, the plant name Plumbagella meracantha. Here, in this plant, in this species, the both haploid and triploid nuclei divide mitotically and to form 4 nucleate type of embryo sac. Out of the 2 haploid nuclei at the micropyle end, one remain as the one-celled haploid egg apparatus. The other moves to the center as haploid polar nucleus. At the chalagal end, one haploid antipodal cells is organized and the other haploid nucleus moves to the center as polar nucleus. One polar nucleus is haploid and other one is the triploid structure of the this one. See, this one is a triploid structure. The structure of the mature embryo sac, the total embryo sac, it is ellipsoidal shape, very thick. The shape is embryo sac is a ellipsoidal shape, thick in multi-layered pectocellulosic cell wall, sometimes to cover by the cutin also. The cell wall is without plasma desmata. Here in the cell wall, the plasma desmatas is also absent. A mature embryo sac is generally a 7-celled structure consisting of one central cell. This is the one central cell, one egg cell, one central cell, and this part is the one egg cells and 2 synergid cells. See, this is the synergid cells, 2 synergid cells and 3 antipodal cells. The total embryo sac, the structure is the ellipsoidal structure, 7-celled structure. In the 7-celled, in the consisting of one central cell. This is called the central cell. One egg cell. See, this is the one egg cell. 2 synergid cells and 3 antipodal cells. The central cell is initially binucleate, but later the nucleus fused to form diploid secondary nucleus. This is the micropyle. This one is the funicle. These are the types of embryo sac. This is the total, the development of the female gametophyte embryo sac. Yeah, the female gametophyte megaspore and female gametotype embryo sac, that is the tetrasporic, monosporic, bisporic and tetrasporic type of embryo sac. These are all the completed. Now we will discuss about to the development of male gametophyte. One second. Yeah. Uh, Now we will discuss about the male gametophyte. Female total is completed. What is female gametophyte? How to develop? In the female gametophyte, what type of embryo sacs are there? What is the monosporic embryo sac? What is the bisporic embryo sac? What is the tetrasporic embryo sac? These all things are completed. Now we will discuss about to the male gametophyte. This is called the microgametogenesis or microsporangiate or microspores or male gametophyte. Microsporogenesis or microgametogenesis, male gametophyte development, formation of pollen grains. First, how to form the pollen grain? In the last class, answer is completed. Now, formation of the pollen grains. Here, microspores represent the being of the male gametophytic generation. Microspore or pollen grains, both are same. Microspores or pollen grains. These two is the beginning of the male gametophyte generation. Older microspore partially after their nuclear formation, nuclear from the tetroid are refused as the pollen grains. During gametogenesis, the pollen nucleus, in the where the pollen is there, in that pollen nucleus divides to form a two cell structure. See, this one is the freshly formed microspore. This one is the microspore. In the center part is called the nucleus. Remaining, this is the microspore, rich in cytoplasm. This is the fresh and rich in cytoplasm with prominent nucleus. This is the very starting micro nucleus, no, rich in cytoplasm. Center nucleus is present, and this is the prominent nuclei is there. In this one, the pollen divide during the gametogenesis, this pollen nucleus divided to form a two cells, and a rapid expansive by uptake lacular fluid. This one undergo two. One is called this is the vacuoles, and unequal divisions are there. After unequal division to form the two cell structure. One is the vegetative cell structure. See, vegetative. This is the large cell. Two unequal of the after this one, these vacuoles to unequal after unequal division to form two cells. One is the large cell, another one is the small cell. There the large cell is there, large vegetative cell, small generative. Where the large cell is there, that is the vegetative cell. Small one is called the generative cell. Generative cell undergo another mitotic division. Vegetative cell undergo to the formation of tube cell. It is not division cell. Only which one cell is divided? Small cell, that too generative cell only. Further divisions are present. Here generative cell, this generative cell may divided generative cell undergoes another mitosis divisions to form two sperm cells, that is called the male gametophyte. The generative cells may be divided inside the pollen grain. The generative cell divided inside the pollen grain or in the pollen tube. Different different species. The development is different types. First one is some are development inside the maybe divide inside the pollen grain. Some generative cell development pollen inside in the pollen tube after pollen has germinated. See this one, this one. This is the critical attached to the wall of the pollen grain, but later comes to lie freely in the cytoplasm of the vegetative cell. See, this is the vegetative cell and generative cell. This one is the generative cell. Immediately after becoming detached from the pollen wall, the generative cell is becoming detached from the pollen wall. The generative cell is spherical shape. What is the shape of generative? Spherical shape. This one is the spherical in shape. After that, it undergoes a lot of extended in the mature pollen grain. It appear in the elongated, probably to facilitate movement into the pollen tube. Synchrony in development within different microsporogenesis is not observed as now. There is no connection between them at the same time, when these changes are going on, the microspore is also synthetic its wall. And how to formation of the sperms. Sperms are formed by a mitotic divisions. How to? Two sperms are formed by the mitotic division in the generative cell. This may taken place while the pollen grains are, this one is the while pollen grains, the pollen grains are confined to the entire is the entire into the entire into that one to develop into the to develop into it may be either it develop into the this way takes place while pollen grain confined to the and another it may be take place after the nuclear of the pollen. In the former condition, the pollen grains are shed at the pollen grains are shed at the three-celled stage. The pollen grains now the three-celled stage, and the later the three-celled stage, and the later and the later at the two-celled stage also. This is the box in tail. And in two-celled stage of the generative cells may be divided inside the pollen grain. After it has a lighted on the stigma or in the pollen tube. before it, sigma in the pollen tube, before it reach the embryo sac. See this one. The outer pollen wall is there. The pollen grain wall of mature pollen grain is stratified. It is compressions are two principal layers. The mature pollen grains contain two layers, the inner layer and outer layer. The inner layer name is called intine, and outer layer, this one, inner layer name is called intine. Outer layer name is called exine. Exine and intine. Exine compresses may be sublayers. Exine. Intine also is pectocellulose presence of beads, ribbon-like structure and enzymatic proteins. Particularly, this is the vicinity of the germ at one. The pollen wall shows various sculpulating patterns are present. After the pollen grains, pollen wall is very thin, called jump pair version where pollen tube come out. The exine, this is a intine. The exine composed of the sporopollenin. This is a proteins. The protein of the sporopollenin to made up of the exine. It is deserved from the carotenoids by oxidative polymerization. The sporopollenin is the extended to the physical and biological decomposition. Because of this property, the pollen grains are often presented for long periods in fossil depositions also. The wall posters pollen grains, this is pulping, and on the exine have crossed taxonomic values. They usually permit the assignment of an individual grain to the family, genus, or rarely to species. The study of the external morphology features of mature pollen grain is referred as called palynology. The study of external morphological features of the mature pollen grain name is called pollen palynology. Uh, the pollen wall material is contributed by the cytoplasm of the spore as well as tapetal cell. Callose wall digested by the calloses. Here in the pollen grains, you can observe to the ubiscope bodies are present. The ubiscope bodies to made up of to the lipids and appear in tapetal cells. Get coated. These tapetal cells get coated by the sporopollenin and are to they formatted for the extended thickening of the exine. The pollen kit is the only layer found on the outside of the mature pollen grains of the many insect pollinated species. Compressive lipids are carotenoids for color from the coating on exine, the pollen tube. This is the actinity as in meat attractive potential pollen grain, the damaging effect of UV radiations, and the main one. The pollen wall can be divided in two layers, exine, intine, inside layer, intine, very smooth. Outer layer is the exine sculpted. The exine again to divide into ectexine, end exine. This again to three parts. One is the ectexine and second one is called end exine. This ectexine again divided into three parts. Tectum, baculum, foot layer. The tectum is the outermost layer. Baculum is the middle one. The last one is the foot layer. This is the exine. See, the exine and nexine. Exine can be divided exine and nexine. This three layer is called this is the intine layer. This part is the intine. This is the exine. This exine again to divided sexine and nexine. That is the exine and nexine. Ectexine and entexine. Ectexine is called the exine. See, this is a sexine. This sexine tectum, columella, baculum. Columella, this layer is called the baculum. This one is the tectum, outermost layer, sexine. Second one is the columella, or this one is called the baculum. And this one is called foot layer. The third one is foot layer. This is called the ectexine, and this total is called ectexine. Another one is the endexine. Nexine, where the nexine is there? Sexine is called ectexine. Ectexine again divide into three parts. Tectum, outer layer. Columella or baculum is middle one. Last one is the foot layer. This is called ectexine or sexine. Next one, nexine. Nexine is the last, this part. After foot layer is part is called nexine. This one is called the endexine. This name is called endexine. Next, last one is the intine. Exine stratification. Morphologically, the exine is divisible into two distinct layers. Outer sculptated layer called sexine by seven inner non-sculptated layer nexine. The sexine further compresses and interned layers of upright rod-like elements are present. The columella, that is called bacula, covered over by a roof-like layer, that is called the tectum. The exine is the tectum may be that typically consists of columella. And is called a tectum. The tectum may be smooth. The tectum may be smooth. Have various types of processing or ectexine, and an inner layer is the endexine. Exine structure. Structurally, the exine is the three basic type. One is a tectum, semitectum, intectum, or atectum. Tectum is the continuous layer is called the tectum. Tectum is a perforated semitectum. Intectate or ectectate, notectum, columella are exposed. Exine sculptated, the exposed surface details of the pollen var. Continuity, the sculptating some of the important types are there. That is the facilate. This is called the smooth. Next, correlated, pitted like structure. Fossilate, grooved structure. Scrabate, very fine projections. Venuclei, wavy structure. Bacula, rod-like structure. Echinate, spine-like structure. These are all the different shapes and size of the pollen grains. Various types, flat, round, ovule, enlarged. Very size is the very smallest mysotist, 10 millimicrograms. 200 millimicrogram cucurbitaceae, Nyctangeraceae pollen grains is the two duplicate one. And next one is the apparatus.
इसका साइज आपको मेजर करके बता रही हूँ कि किसी में M40 होता है, दैट इज बता मैं किसी में M40 होता है, किसी में M38 होता है, जिसमें साइज होगा मैं आपको यही पे डिफरेंट लाइन की मेजर करके बताऊंगी दैट देयर वुड बी एनी कंफ्यूजन आप बिल्कुल बिंदास करा सकते हैं। ये हमारा जा रहा है 38, इसका M is 38, so that means हमारे पास साइजेस इसमें है 38 and 14, 38 and 44, 12,
B carrier on pollen surface, which is directly individual associated with it is generation long apparatus is called polpi or pair-like parrot. Another one is the very important NPC system. NPC meaning uh, refer to the number, position and character system. This is the pollen grain number, pollen grain position, pollen grain characters. That is called the NPC systems of the operator. This system to
इसमें आपको ये प्राइस मिलेगा 1350, 40, 44, 46, डिजाइन नंबर,
this NPC, a system classify pollen grains on the basis of operators. For example, pollen grains without operators, that is the no. Monohemi N1, ditrimid N2, poly minimum. Position operator, category and anapton when the prominent digital. This is the total development of the pollen grains also. Female gametophyte. Thank you.